space over here is called the intermembrane space. Okay? I'm going to simplify it to just put IMS, yeah, intermembrane space, short format. Therefore, if this is the intermembrane space, and this is the matrix, and I've zoomed in, this membrane is the it's the inner membrane. Okay, so be very adaptable to look at it from a zoom out perspective as well as a zoomed in perspective. Okay, so matrix, intermembrane space, the inner mitochondrial membrane. Don't be surprised if we flip things around. Like if someone were to say this is the matrix, then this is the inner membrane, then this will have to be the intermembrane space. Okay? So we may throw you off just by turning the whole diagram on its ends. And therefore, if this is the inner membrane, there must be an outer membrane somewhere here. Yeah? So I do not have one more layer of membrane to paste up here, but this is the next layer of membrane. Believe it or not, I used to draw this out and it took ages, so I decided to make magnets out of them. Okay, so this is the outer membrane. Therefore, out here would be the thank you, cytosol or cytoplasm. Some of you have asked me what's the difference between cytosol and cytoplasm. At least for us, right, we don't quite differentiate between the two. But if you really go and look into the nitty-gritty of the difference between the two, cytoplasm is often defined as the jelly-like substance plus everything you can find inside. All the nutrients, all the organelles floating around. Cytosol refers to just the jelly-like substance. But we're okay if we use either or. Yeah? Right, so that being said, let's recap the 4 steps of cellular respiration. We begin first with glycolysis. Okay. Glycolysis. In glycolysis, a quick recap, we convert glucose to pyruvate. Okay? But some other products are also produced in the process. We get a little bit of ATP, we get a little bit of water, but one thing's for certain at every single step. We generate this one fella, right? NAD plus as an electron carrier, take out some electrons, and then you get NADH. Yeah? Mind you that whatever I do here, often it's not chemistry accurate. Like it's not balanced in any way. Okay? But if you want a balanced equation, FYI, it will look something like that. NAD plus 2 electrons plus H plus, then you get NADH. Yeah, this is the balanced form. I do not require you to know this. Okay, I have obviously simplified it to look something like that. Okay, because that's really what I'm focusing on. And this NAD plus can take electrons. I quite like last week one group called this nada. Gosh, I've heard nada for now. Then there's a father, right? There is nada and father. Okay, I'm gonna use some magnets along the way because I'm tired of drawing. Okay, so NAD plus, at the end of the day, if it receives electrons, you get NADH. Along the way, I'm going to be adding on electrons in the form of blue magnets. Yeah? Plug, plug. Okay, so yes, gain electrons. Electron carrier, yeah? Right. So from here, next step takes place in the matrix. Yeah? The link reaction. This is short form, but don't copy me, okay? Spell out reaction. Okay, the link reaction takes place in the matrix. What happens there? Pyruvate, you end up getting acetyl CoA. You also get some other byproducts like CO2 along the way. Yeah? But again, one thing's for certain, you will use up your electron carrier, grab out some electrons, you get NADH in the process. Okay, let me add on to that. now CoA will then be used for the next step. 3, the Krebs cycle. In the Krebs cycle, A star CoA is used. A lot of things are generated in the process. One thing is common, you end up generating NADH again. Plus, you get NADH, yeah? But now you will encounter further. Okay, this is also an electron carrier. Other than that, we know that a little bit of CO2 is produced. We know that a little bit of ATP is produced. And this being the Krebs cycle. We just add some more others. Another. What is a father? Father. Right? And then we should pull out some more electrons, yeah? Feel all okay? It's just the first 3 steps. Again, I do not need you to know the details of the reaction. Just be familiar with the products and what is required. Because if you notice, at every step, the product seems to be required for the next step. Yeah, the product here is required for the next step. Okay. On top of that, another thing to take note of, at every step to generate NADH, or at least a reduced form of the electron carrier. And we've been doing all this for this last final step. You have questions for me, anyone? Yes?
So like is the pyruvate like transported from the cytosol to the matrix?
Right, okay. There seems to be a disconnect here. Pyruvate here, but we need it for late reaction. So yes, indeed, it will need to be transported through the intermembrane space into the matrix for it to carry out the reaction. Yes? For 1, 2, 3, 4. What's 1, 2, 4? This one over here, is it? Okay. Are these proteins enzymes? If I'm not wrong, the first one is an enzyme that helps in the extraction of the electron, yeah, from NADH. I do not recall the rest being enzymes. There may be, but I may be wrong in this aspect. Oh, I see. Do these fellas be considered coenzymes? I've not heard them being called coenzymes. I suspect we call it coenzymes when they directly interact with each other. So there's no direct interaction per se. Correct. So I think it's called coenzyme Q because it interacts with other proteins. Some of them are enzymes in the process. Yeah. At least from what I've observed, a lot of times you call something a coenzyme, it will interact with it or bond with it. Interact with it closely for at least a period of time. Yeah. So why have we been doing this first, second, and third step? Well, and why have I been using magnets? Okay, so that I can move things around. Right? We've been generating lots of NADH because now let's use them for a meaningful purpose. Okay, we'll take the NADH from there. I think it's not fit, okay? Then we have some NADH here, we have some FADH2 here, we have some NADH from link reaction. Let's channel all of this to the last step. Yeah? This is the very last step. We've been collecting lots of electrons. For what purpose? So that we can channel it down this chain of proteins called the electron transport chain. Chain of proteins transporting electrons. Okay? You may find this in your textbook. that diagram that doesn't quite move much. I'm going to now use other magnets on the whiteboard. I'm going to use pink magnets to represent the protons or the H plus ions. Okay? Right, so what happens here? Well, this happens. This happens. Yeah, I have oxygen at the very end. This will serve as my final electron acceptor. Okay? So if I simplify the whole process, it goes something like that. As NADH gives up its electrons to the electron transport chain, we regenerate NAD plus in the process. Yeah? I'll show again. Yeah, I put a lot of work to do this magnet. I'll show you. Okay, so NADH, when it donates its electrons to the electron transport chain, then it will regenerate NAD plus in the process. Yeah? Regenerate for what? Don't waste it, let's recycle it. So it can be reused in a link reaction, for example. Okay? We pass it down the chain, but every time we pass electrons from one entity to another, a little bit of energy is released. And we use that energy to actively transport protons from the matrix into the intermembrane space. Every time we transfer electrons, some protons are actively pumped from the matrix to the intermembrane space. Last but not least, these electrons need to have a final destination, and its final destination is oxygen. When oxygen receives the electrons plus a little bit of hydrogen ions, we end up getting water. That's why the breath we breathe out contains not only carbon dioxide that came from Krebs Cycle, that came from the link reaction, it is also wet and moist, the breath that we breathe out. So actually, if we look at the broad steps, now if someone were to ask you, at which step is CO2 produced? Right, the CO2 that you breathe out. Actually, very specifically, is produced at step 2 and step 3. When is oxygen really needed in cellular respiration? Well, actually, only in the last step, not quite the first, second, and third. Right? So now when you look at the equation of cellular respiration, you actually know which component is used at which step, which component is produced at which step. Last but not least, why have we been generating this high concentration of proton versus a low? Right? The word you can use in your vocab is called proton gradient. The idea of a proton gradient is that there's a high concentration in a low concentration area. We have created a proton gradient, if you see on the whiteboard. There is a high concentration of H plus ions, I, inside the intermembrane space, and then there's low concentration in the matrix. That's why we say there's a proton gradient that has been set up. But why have we been doing this for this final last step? Because when these protons accumulate here, now they want to flow back down via facilitated diffusion. So they start to flow back down. And as they flow back down into the matrix, ATP synthase, this enzyme, will finally get to work. I'll now show you a video of this happening so that you can appreciate why we need the protons to flow through the enzyme. This is the enzyme up close. The protons are represented by the green cubes that you will see moving.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP.
It is a very old documentary. You see the protons coming in?
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized.
This is the protein structure, right? So don't be surprised if we show you this structure and ask you, what level of structure do you see? Okay, what level of structure do you see? Okay, up to quaternary. This protein is huge. It has up to a quaternary structure. Can you see a lot of alpha helices? Yeah? So at every chapter, there's always room to link back to the previous one. And you can always see ATP and ATP going in. Then pop, like you top up credit card or EasyLink card, and then after you can take it out of the ATM machine. You top up, you pull out your card. Okay, so that is a little glimpse of how ATP synthase works and why we require the ions to flow through it. You do not need to know the details at all. I'm just showing it to you so that you can appreciate it. Because students always wonder, so why do we need to flow through? This is why we need to flow through. Almost like if you see rivers with wheels attached to a river, right? When the river stream flows through, you see the wheels spin as it catches the water. The ions are almost doing exactly the same thing. Yeah. ATP synthase is catching the flow of the ions going through, and that helps to rotate parts of the enzyme. Okay? So, to sum things up, this is what it looks like broadly. Okay, we tie everything together. Here it is. Okay, this is the last step.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
And that ties up the very last step of cellular respiration. This is the toughest part of this chapter. If you found it understandable, then I would say this chapter is quite good for you. Okay? And it will also help you in the next chapter, because in photosynthesis, you'll see something very similar happening. After all, if you notice the equation for cellular respiration looks almost like a reverse of photosynthesis. We will go reverse in a while. Okay, I give you some time now to take down some notes or ask questions if you have any. Yes, Kaisin? Okay, if the NAD plus, is there ever a need to produce oxygen? Oh. Okay, why would you ask if there's a need to destroy or to the NAD plus? Yeah, that's why we use the word regenerate, because we don't necessarily whatever electrons that went into the electron transport chain, we get to regenerate NAD plus. Then it can be reused. It can be reused here. It could be reused here. So you're asking, should we destroy some? Okay, that I don't know. I don't know if there's a set amount. What is the ideal concentration for any organism to live? But if I refer to any biological system in the body, there tends to be a range of amounts that's most ideal for any reaction to occur. Every time we have too much of something, the body will proceed to destroy the excess. That's usually how our biological system keeps a balance. Okay. Yes, Xuan? Okay, what's chemiosmosis? You know in your textbook, you see the word chemiosmosis? Chemiosmosis is just a very complicated way of saying a flow of protons from the intermembrane space back into the matrix. Chemi is like chemical, osmosis is like the flow of something, right? So chemiosmosis. It is like chemiosmosis would be a verb, therefore. It's a process. Yes? Oh, what stops the protons from, rather than go through here, right? Just go through here like that. Okay, great question. So therefore, let's tie this back to the chapter of membranes. Okay, when we look at membranes, we know that the membrane is made out of phospholipids. Therefore, it has a hydrophobic core. Great question. So if water can pass through, why can't an ion? Okay, then we look at the property of water and ion. When we look at H2O, H2O is polar. An ion like H plus is fully charged. Okay, so that's the first thing. If we compare the two, one of them is a lot more charged. Right? Number two, you're right to say this is much smaller than a water molecule. But I don't know if Mr. Low taught you this. When you have an ion, that means to say it can be surrounded by water, right? That's how ions can stay out as an ion, right? So if I were to join the water molecules in H2O, so it would be like that, right? Am I right? Yeah? So actually, although the ion itself is small, it becomes much bigger when it's surrounded by all these water molecules. So it doesn't pass through anymore by itself. Questions? The part that I wanted to study, you to study yourself, maybe I'll just quickly run through so that we have a little an idea of what you're going to encounter. What if you do not have enough oxygen in your body? Okay? What are the implications if you do not have enough oxygen in your body? Can you see where the jam will be? Can you see where things will clog up? Can you see where things will grind to a halt? If I do not have enough oxygen? That's the conclusion. For one, if you do not have enough oxygen, now I'm missing my final electron acceptor. NADH can't quite pass its electrons to the last step anymore. Yeah? If I do not have enough oxygen. If I cannot pass this down the electron transport chain, I do not have NAD plus regenerated. Do you see the number of steps that require NAD plus? Lots of steps require NAD plus. If I'm unable to regenerate it by giving up electrons, a lot of the previous steps will start to grind to a halt. And so, sometimes people will say, oh, Krebs cycle requires oxygen. It requires it indirectly. Because if the oxygen is not present in the last step, the previous steps will all start to grind to a halt also. Okay? Simply because I will start to run out of NAD+. I need it to be regenerated so that I can use it in previous steps. So we have a problem, right? Everyone has encountered a situation where you've been, somebody cover your mouth and don't let you breathe, right? No? Or is it just me? Okay. No lah. Okay, I'm sure you've encountered a situation where you were asked to sprint around the track, right? And then you start to get cramps everywhere, right? Your legs start to cramp up. Or if right now I ask you to compete among yourself, you raise your hand, then let's see who can last the longest. You do this. Just keep doing this, and then we see who can last the longest. After a while, you start to feel pain running through your arm. That pain is a result of a chemical that's being produced. And you've heard of this chemical produced before. Lactic acid. Heard of that before? Yeah? Lactic acid. It's what results in the pain that you feel in your body when we run out of oxygen, or rather when there's not enough oxygen present. Let me just complete the process here. NAD plus, NADH. So our body has a way of coping. Glycolysis, remember, generates a little bit of ATP. In the event that you do not have enough oxygen in your body to meet the supply of ATP that you need to continue that sprint, our body has a backup plan. The backup plan is to make use of glycolysis to give you just a little bit, squeeze out a little bit more ATP for you. It does it by doing this. The pyruvate that is produced, rather than bringing it all the way in for the link reaction, it channels it to produce lactic acid. But what's so special about this process? Well, for lactic acid to be formed from pyruvate, it needs electrons from NADH. So it will take the electrons from NADH. But you know what happens in the process? You regenerate NAD+. So the electron that was taken away from glucose will be given to lactic acid inside. But if I keep recycling this process over and over again, I could produce a little bit of ATP to keep you going a little bit. But it won't last very long. You start to feel the pain running through your body. That is the body's way to get your whole body to stop doing what you're doing. Because you will need oxygen after a certain point. We call this process anaerobic respiration. Na means don't have, aerobic means like breathing. No aerobic exercise. Yeah, anaerobic, no oxygen. You know what yeast is? Yeast can do the same thing. It can also carry out anaerobic respiration, except the products are a bit different. Rather than produce lactic acid, it produces ethanol and alcohol, as well as CO2 in the process. And guess what? We make use of yeast to create a very famous alcoholic beverage, like wine, like beer. Brewers just toss yeast into a vat of grape juice or malt juice, and then they seal it up, deprive the yeast in there of oxygen. Then the yeast inside the pool of sugars, that wine and beer, the grape juice contain glucose, right? In a sealed environment without oxygen, they will start to produce alcohol and carbon dioxide. That's why beer, wine, they are a little bit bubbly, they contain alcohol. This does not take place in human beings, mind you. If not, that means I ask you to go for a run, you hold your breath right, you start to get more drunk as the time goes by. You also do not produce CO2 in our version of the process. Okay? We just produce lactic acid. Yes? Right, okay, so trying to ask, if after the run, then you stop there, and then this is what you all do, you like, right, you catch your breath, right, you top up your oxygen, right? Okay. All the lactic acid that was running through your body, it will go through a reaction with the oxygen that you just breathed in to reform pyruvate, if I'm not wrong, if my memory recalls correctly, so that we can continue the process onwards. Okay? We call that replenishing your oxygen depth. Okay, so that's a little bit of anaerobic respiration for you. I found out that there is an animal that goes through a form of anaerobic respiration that will produce alcohol, and that is a mighty little goldfish. So the goldfish, right, if you deprive it of oxygen, right, its alcohol content in the blood will start to increase. I did Google before, like, if you want to make an alcoholic drink out of goldfish, like put it in wine, you know, maybe. Like, how many goldfish do you need? Like, how long do you need to deprive the goldfish of oxygen? It's quite long. Can be deprived in very, very long periods of time. Okay, and we'll end off with the mitochondria. We'll do a 2-minute mental break and mentally pause and consolidate, take down some notes, take a mental break for yourself. Yes. Okay, Ishan asked, how does pyruvate become lactic acid? Just a chemical reaction, right? But in one sense, I could say that from pyruvate converting to lactic acid, we are reducing pyruvate. Yeah? We are adding electrons to pyruvate and it converts into lactic acid process. We are reducing pyruvate. From glucose to pyruvate, we were oxidizing glucose. We're taking electrons out. You can find videos to complement your learning in Lumos. All these videos are no longer than, I try to find short videos, they are very succinct. Each one usually doesn't stretch beyond like around 5 minutes plus, but it gives a nice summary of every single step. If you need a certain steps to recap, you just search for glycolysis. If you need to know a little bit about NAD class, you could search, it will be all over there. Okay, so a quick revision would be to watch all these videos. Yeah? You can find videos I show in class also, right? So it should be all consolidated. And if you don't know by now, I no longer update the class Google Classroom every time we record a video. You will instead go to, where will you go to? You'll go to the main page and you should find all our recordings there in case you want to search for them and in case you have not navigated before. When you go to 302, you should find the most updated video there. Okay? Because I will not put out Google Classroom anymore. Very tiring. Okay? Alright. We all just take a mental break or restart by all. Okay, so that you don't feel insecure about this. If you look at question 3 of your CA, what else is needed for oxidative phosphorylation to occur? Okay, I hope you managed to figure out what is required. Things that are required at this step will be things like NADH for it to occur. We will need oxygen at the end as the final electron acceptor. Oh, yes, thank you. Who said that? Okay. You need the proteins of the electron transport chain. Proteins of the ETC. You may say ATP synthase, the enzyme. Okay? Okay, some of you may say, oh, protons are needed, right? But the protons don't exactly participate in the reaction. It's the movement of it that helps it, the things happening. Where do these components come from? Oh, that's a good question. Where does NADH come from? Okay, it came from your previous steps. Where did oxygen come from? Okay, you have to breathe it in. Okay. Where do these proteins come from? Or ATP synthase? Okay, yeah, if you really must, they are proteins. Therefore, they must have been synthesized by ribosomes. Okay? Question 4. They ask you to name the process used to transport the H plus from one region to another. Okay, there are two processes here. First one, protons being transported to the matrix, from the matrix to the intermembrane space. This is active transport. When it flows back down, that is facilitated diffusion. Okay? One requires a gradient, the other sets up the gradient. Okay, so two processes. You will not find this in the textbook in this part of the notes because these are links being drawn back to previous topics. Right. Okay, so far in cellular respiration, we went in this direction. We had glucose, and we needed oxygen, and then we produce water, not pyruvic, right? We produce CO2, we produce water, and we also get ATP in the process. Yeah, it's a broad equation. We essentially have been transferring electrons to oxygen, except we break it up into several steps, and we require NAD+. In photosynthesis, you learn in primary school, it's a process where we produce sugars. If you look at the equation of photosynthesis, it looks like the opposite direction. Right? Photosynthesis, we require CO2, we require water, we also require sunlight and chlorophyll. What do we get in return? We get glucose and oxygen. We spend so much time on this in cellular respiration because once you get this, photosynthesis is kind of the reverse. Okay? So I want you to pay close attention. At what point is CO2 used? At what point is water used? At what point is glucose produced or oxygen produced? Right, try to spot it along the way. But before we dive into that, we need to understand this part a little bit more. Okay? So let's first dive into the structure of the chloroplast. We started off with the structure of the mitochondria. Your lesson notes, I will be referring to page 41 and 42. You're wondering what I'm referring to. Most of the times, the things I require you to highlight have been bolded already. Okay? I'm going through this so that you have perspective. Every time I start bringing up a part, you know what we're talking about. Alright, first and foremost, in a while's time, I'll need a volunteer to come out and do a demonstration with me. Okay, first and foremost, chloroplast, just like the mitochondria, has an outer membrane. Okay? It also has an inner membrane, except its inner membrane is not highly folded at all. This is where chloroplasts are special. It has a third layer of membrane. This third layer of membrane is highly folded, but highly folded into very unique shapes. If you look into your textbook, they fold themselves into these pancake-looking structures that stack on top of each other. Over here. Okay, so this is a cross section of it, right? If I look at the side view, you can sort of see the last layer of membrane used to form small little discs like M&Ms. Okay, there are lots of these M&Ms. Okay, so let's put a name to all these M&Ms. Huh? You can refer to these M&Ms as thylakoid, right? The membrane that it is made up of is called the thylakoid membrane. The space inside the thylakoid membrane is called the thylakoid. In your textbook, they wrote it as lumen. Or if you write thylakoid space, that's also okay. And then there is the fluid or the space surrounding all these thylakoids. We refer to that space as the stroma. Right? And these are kind of the new structures and places you'll be looking at for this chapter. A lot of the reactions take place at these areas. Anything to clarify? Yes, hi. Right. I believe a stack of them is called granum. Yeah? But and we rarely make use of the word granum. But you could, there's nothing wrong with that. It's just a whole stack of these M&Ms. If you ever have the privilege of looking at one of these under the microscope, they look like flattened pancakes. If you look under the microscope, you see inside the chloroplast, you see all these stacks over here. These are all thylakoids. Okay, so you see my diagram there, it's like totally different now. Alright, very compact. This one is like flattened out. Okay, but many stacks. Almost look like RER, minus all the ribosomes. Where do you find chloroplasts? In plants. Okay, so here's a question now. You can find mitochondria in plants. Okay, a plant cell. Huh? Yes. So plant cells are also have mitochondria. Maybe wondering what for? Okay, this is what it's for. Remember the two equations I've written, right? Okay. If the chloroplast role is to produce glucose, then the mitochondria will make use of the glucose so that it can produce ATP. Plants also need energy. The only difference is that this glucose is produced by itself, whereas we have to eat the plants to get the glucose. Okay? Yeah, so it's a trick question. Often students think plants only have chloroplasts, don't need mitochondria. But plants only have chloroplasts, it will just produce lots of sugars. It has nothing, it cannot do anything with the sugars. Right, so it has both. Does the chloroplast have intermembrane space? Have, but it's not relevant for this chapter. Okay? Does it have an outer intermembrane? Also have, but it's not that relevant, you will find. We will mainly refer to this membrane a lot. Okay, so 1, 2, and 3 are the ones I'll refer to the most. You'll find me talking very little about the inner and outer membrane. So, where do you think we can find chlorophyll in that mess of a structure? Okay, where do you think chlorophyll is found? Inside? Okay, it's found inside, okay, right? Where inside? Okay, it's not free floating around. Okay, inside the thylakoids? Actually, it's on the membrane. So you find the chlorophyll pigments, right? You'll find them on the thylakoid membrane. That's actually where you find it. Here, I've extracted a whole jar of chlorophyll. Yeah? So, not too hard, just bleach the plant, soak in alcohol. Okay, I soak the bleached plant in alcohol because alcohol is amphithetic. Because it's amphithetic, it can break down the phospholipid walls of the chloroplast. And therefore, all the chlorophyll will come out. So this is how I extracted it. You can do it at home yourself if you want to. Really, I'm not joking. Just go take a blender. If you have vodka for some reason, you pour it in. Then you throw, I use pandan leaf, I throw pandan in, then after that, you get it lah, but you have to filter it. Okay? Sorry? What do I do with this? I use it for science. Okay. So now let's move on to the next part of our lesson. Let's talk a little bit about chlorophyll. Why do plants have chlorophyll? What's so special about chlorophyll? After all, in primary school we know, or at least all we've been told is that chlorophyll can absorb sunlight to make food. But whatever does that mean? So let's look a little bit in that today. For that, I'm going to go into CA 3.5 and I'll begin first with activity number 1. Now I need a demonstration partner. Anyone wants to? Oh, Zoe, you're volunteer? Okay, yes.
Is the thylakoid membrane made out of
Yes. Any membrane you find in a cell is made up of phospholipid bilayer. Okay? That's why you also, when you use hand sanitizer, there's alcohol inside. The alcohol is amphithetic, can break down the phospholipid bilayer of, let's say, a bacteria to destroy it. The coronavirus also has a phospholipid bilayer on its surface. Use ethanol on it, and then the membrane will just come apart. Yes? Chlorophyll is found on the membrane. Thylakoid membrane. No, not on the outer or inner, just on the thylakoid. Okay, this is where you find chlorophyll on the membrane itself. We'll look at it in a while. Yes. Oh, so are you saying is there a difference in the content of the inside the thylakoid lumen and the stroma? Is that a question? Right. So is there a difference in the content here and here? Yes, there will be a difference in the substances you find inside. Why I would say that is because, hey, shucks, did anyone start the recording? Okay, thank you. Okay, so I say that likely there will be a difference because certain reactions take place here and not out here. So therefore, I can expect that there will be some products here that's formed that will not find out here. Or some chemicals that's needed for this reaction that's found here but not out here. So that's how I will conclude that definitely the contents here and out here in the cytoplasm is slightly different. What? Oh, what is the main fluid? Is it? Okay. Anything fluid in our body is water. Okay? Water is the best universal solvent in our body. Maybe in an alien planet, maybe it's oil. Yeah. That would be quite interesting, right? Then the phospholipid bilayer, everything will be flipped the other way around, right? Yeah. So at least on Earth, it's all water. Okay, to understand a little bit about chlorophyll, you now need to learn a little bit of chemistry, which I'm very bad at, so I'm giving a very simplified version. And Mr. Low, if he watches, will cough blood. Okay, some of you already know this because you are presenting this for science particles. Okay, so you know that there are many models of an atom, right? I'm going to show you one model. You've seen this model before, right? Let's say they are orbitals. Okay, and then atoms or nucleus, right? The red dots here represent electrons. I don't know what atom this is. I'm just feeling it as an example. I know some atoms have 4 electrons. I also don't know what this is, but just an example. Okay? Don't read too much into it. But why I'm showing you this is because of this. We know the electrons exist at certain orbitals because they have a certain amount of energy. Correct? Okay, correct. Yeah? Electrons exist at certain orbitals because they have a certain amount of energy. Yes, Satosh? Okay. Okay, but that's the thing. Actually, we can excite electrons. If we can just supply electrons with energy, there is a way that we can excite electrons from one orbital to a higher energy orbital. Okay, there is a way. And I would say that this electron that has just jumped is now excited. It is in its excited state. Oh wait, did Mr. Lo teach this? Kind of. For what? What topic is this? Topic 1. Okay, atomic structure. And therefore, anything that's excited can go back to its ground state. And here's the interesting thing. Energy that was supplied to cause this electron to be excited, when it goes back to its ground state, actually it also releases the energy. Okay? But it can release the energy in various forms. It could be thermal energy. It could be light energy. As I was going through NQC, I found a drink that, if you look at its ingredients, it says tonic water. It says it contains quinine. Quinine happens to be a chemical that, when supplied with enough energy, you could excite its electrons to its excited state. But when it goes back down to its ground state, it will release the energy that I just supplied. So, right now, I'm going to show you what happens when I supply with enough energy. Here, I bought a high-powered UV gun. Yeah, not a UV gun, it's just a torch that can shine UV rays. You don't quite see anything coming out, right? Because UV light is invisible. There is no color in UV light because it is high energy, right? So, I'm going to supply the chemicals in that drink with high energy UV light. Do not shine this in your face or your hand because it's high energy UV. UV is cancerous. Every time I do this demo and show on my skin, right, after the lesson my skin will start to itch. But may I get a volunteer? Sorry, I should have said that after. Anyone would like to volunteer for this demonstration? You'll be perfectly safe. Maybe like 90% safe. Yeah. Alright, ready? Okay, Isabel, you really volunteer yourself? Okay, thank you, Isabel. Let's give Isabel a round of applause. Let's exercise her hand. Yeah, come on. Hey, you take off your shoe, very comfortable. Okay, so UV, B, strong energy, no, a lot of energy in these rays. Alright, first and foremost, let's pour a little bit of this drink out. You can keep this drink in. Okay? You do the honors. Okay. All good? All good. Okay, so Isabel, pour a little bit of this liquid out so you can see what it looks like. Wait. Okay, we'll see what it looks like prior to supplying it with energy, okay? Okay, can I? Very good. Okay? Nothing special. It's just like plain water, yeah? So if your parents drink this drink called gin and tonic, it is what is the tonic part. Yeah? So this can part is the tonic part. Alright, so as you can see, without supplying it with any energy, this is what it looks like. Now I'm going to supply with energy. Isabel, I'd like you to do the same but aim better. Aim it into this glass bar. I'd like you to pour it into this glass bar. Ready? 4. Yeah? Right now, I'm supplying high energy rays into the electrons. Electrons being excited as it goes. And as it's being excited, it's also going back to ground state. So it's a continuous process. But the energy that's being released in this case is light. If I stop supplying it with this high energy, you don't quite see the same thing anymore. Right? Supply it back, you see the same thing. Isabel, thank you so much. You can keep the tonic water. Okay? There are a lot of other things that can, okay, the word here is fluoresce, right? The idea of fluorescence is that you supply with high energy, the electrons get excited, go back to ground state, you release light in the process. There are a lot of things that fluoresce in the classroom. For example, the paper on this wall also contains chemical that allows it to fluoresce. Okay? But if I move beyond the paper, you don't quite see anything. Right? But in the paper, the electrons get excited, going back to ground state. Other things that fluoresce. If you have highlighter with you, and if anyone highlights a page of okay. Yeah? In the name of science. Okay. Okay, so yeah. Okay. So a lot of things can fluoresce. What about the chlorophyll that I've got here? So here I have about chlorophyll. Let me guess what color your fluoresce. What it just absorb light, right? After all, plants make use of the energy to after that synthesize sugars at the end of the day, right? Okay, these chlorophyll are a bit different in the sense that I pulled them out of the systems that it was part of. It's just free, right? I'm going to shine light right now. I'm going to make a guess where it turn. What? Light? Color. Ready, huh? You see? It doesn't fluoresce as brightly as quinine. You see? But you can see that it's kind of like a blood ring. Right? I can assure you, later on if you come to the front, it is a deep green. Right? Chlorophyll fluoresces ray when you supply with enough energy. The electrons can be excited, and when it goes back to its ground state, we release the energy in the form of red light. That's what chlorophyll can do. But do you see the leaves out there red in color? You don't. It means to say something. The energy must be used for something more meaningful, not quite shit up into the sky as red light. Yes? Sorry? Oh, does it become hot? Not that I'm aware of. It doesn't become hot. Yeah. So, but did he say it become hot? A little bit of thermal energy. Yeah. Okay, so actually there's a page. If you spoiler alert yourself, there was a page in the textbook that actually said it will be red color. That's the what? Well, what kind of energy? I don't know. Anything? I mean, I call it light energy. But I guess the energy is stored in the waves. You all learn light yet? Light is about photons moving along a wave. So there's energy stored in the photons that are being transmitted along the waves. Yes? What's the difference between black light and UV light? Black light and UV light are the same. So here I can call this black light also. Yeah. Another name for UV light. In the light, in sunlight, there's both UV light. There's also colors of the rainbow, all those light. Oh yes, it is. That's why you must put sunblock. Yeah. That's why you go out into the sun for prolonged periods of time. Make sure you put sunblock, so that it can protect you from the UV light.
Maybe you just wondered what fluorescent means. We need to look at energy and atomic structure to explain this. You may recall that electrons are found around the nucleus in discrete and well are not constant, but become smaller as you move away from the nucleus. Electrons may move from lower to higher energy levels if they receive an amount of energy exactly equal to the size of the energy gap. This is to do with quantum theory, which you may wish to research more by yourself later. The energy needed is easily transferred to the electron in the atom by a suitable part of the electromagnetic spectrum. Let us look at one such interaction that leads to an electron transition between energy levels. When exposed to a certain wavelength of light, an electron can move from a lower to a higher energy level with the atom. We say that it has changed from a ground state to an excited state. This excited state is normally very short-lived, and the electron will fall back down, returning to the ground state. But in doing so, must emit the same wavelength of light that it had absorbed earlier. Let's use a more simple way of showing these transitions by drawing only the energy levels themselves as straight lines and not circles. Now we add another energy level. In this example, the electron has moved from the bottom to an energy level 2 levels higher, and then returned again to ground state. The atom will have absorbed and then re-emitted the same wavelength of light. But what if it doesn't fall back to ground state in one step? Here, there is a 2-step return to the ground state, and so the atom emits 2 different wavelengths of light, each with a longer wavelength than originally absorbed by the atom. This is fluorescence. If the absorbed electromagnetic radiation is in the invisible ultraviolet part of the spectrum, or short wavelength, then the light emitted will be of a longer wavelength and be in the visible part of the spectrum. If tonic water that contains quinine is exposed to UV light, it glows with a blue colour. This graph shows the wavelength of UV that quinine absorbs, mainly around 350 nanometers. The excited electrons in quinine return to their ground state in stages, emitting a longer wavelength of light, mainly around 460 nanometers, the blue region of the visible spectrum.
Okay. This is more of FYI, so that you can appreciate what's happening in the plants. I do not require you to be able to explain this because they are bio. Okay? Alright, so now with that piece of knowledge, let's look at a little bit about chlorophyll a little bit more. Okay? First and foremost, we need to know the where. Where do you find chlorophyll? Chlorophyll is found not free floating. Chlorophyll is also not embedded. Not just embedded like that in the membrane or the thylakoid membrane is not. It's actually found within protein structures. Right? And the protein structure looks something like that in your notes. You go to your notes and you refer to this page, it looks rather complicated. But actually it's not that bad. You look at this diagram here. Okay, this diagram shows a cross section of this protein. Right? I'm going to draw on the whiteboard now, the protein. First and foremost, you see the light purple part in the middle? That's one part of the protein. So I'm going to represent it by this cylinder. Okay? This light purple part, we call it the reaction center complex. The word complex often means that it's made up of many parts. So even within here, understand that there are a lot of different things that make up this structure. Okay? Next, surrounding the reaction center complex are many protein structures, and it's kind of like an orange, all the segments of orange surrounding the core. So if I were to now draw the segments surrounding it, I could have one segment be I, and then one segment over here, and then maybe one segment down front, another segment over here. So you see what's happening? Actually, I have all these segments will surround the reaction center complex, like an orange. I refer to each one of these segments together, these two things, these two components, make up what we call a photosystem. Photosystem. So where do you find chlorophyll? In your textbook, you see those green pigment molecules. The bottom right hand corner, they label it as pigment molecules. Okay, they are referring to chlorophyll. Okay, so maybe you want to write down chlorophyll over here. Because when students read this, they are wondering what pigment molecules are. It's referring to chlorophyll. And that's why they color coded it green. So where do you find chlorophyll? Okay, I'm going to slice this photosystem in half now, which is what you see in this diagram. Okay, let me slice it. Slice the photosystem open. When you slice it open, you will see the reaction center complex in the middle, and you will see the light harvesting complexes at the side. This is what it looks like when you slice it in half. So where do you find chlorophyll? You find them actually planted within the light harvesting complexes. Right? And that's what all the green circles inside are. You will also find a very special chlorophyll molecule at the base of the reaction center. Okay? So this is where you can find chlorophyll pigments. Okay. So you see, I'm going to come to the point of why we learn about excitation and ground state, right? Well, chlorophyll pigments have electrons that can be excited. So let me now, let's refer to this diagram now over here. Okay, this is the same image as what's over here. Same up. Okay. You see these white circles? Okay, I'm using them to represent the chlorophyll pigments. This circle here also a chlorophyll pigment. It's the same thing as what you're seeing here and on the screen. Except now, let me put the electrons in so that we can play with the excited electrons. I'm going to put the electrons into these chlorophyll pigments. Oh, there's one there. Alright, great. So we have electrons inside the photosystems now, the chlorophyll pigments. Right, so what happens when light strikes? What happens when light strikes the light harvesting complex? Okay, this is what happens. Okay, they're going to get excited. This is how I'm going to represent excitation. It may look very funny, but just bear with it. Okay, so light strikes the light harvesting complex, the electrons get excited. I'm going to show you how it gets excited. When it's excited, it jump out of the page. Okay, so it's this excited. Okay, so like ready, so boom, then after that we'll go back to ground state, right? Okay, so that's how I represent excitation. Okay, but where does energy go, right? Excite, go back to ground state, but energy must be lost somewhere, right? Okay, but it's not, right? That's why we call it the light harvesting complex. Because after the first set of electrons get excited and go back to ground state, the energy that is lost is gained by the adjacent electrons below. So, kind of like a wave. So what now? Excite, but as it goes down to ground state, the next set of electrons get excited. As it goes back to ground state, the next set of electrons get excited. As it goes back to ground state, the next set of electrons get excited. You've been to a match before where people do like Kallang Wave. Yeah, same thing. Right? You all just pass the energy along. Yourself as a chlorophyll, you don't jump to the next seat, right? Instead, you just pass the energy along as a wave. Right? So that's really what's happening. Still okay? Alright, but here's the interesting part. Remember the electron transport chain in cellular respiration? We needed NADH to contribute electrons, yeah? Yeah, in cellular respiration, NADH was what contributed the electrons to the electron transport chain. In plants, the thing that contributes the electron is not NADH, it is this protein structure. Okay, so I need this protein structure to now just ship out one electron so that it can go down the electron transport chain. Right? Okay, so how does this happen? It happens like that. Because if enough energy is harnessed harvested. As this goes down to ground state, now this electron over here will harvest all the energy of the surrounding light harvesting complex. Imagine everything concentrated into this electron over here. You gain so much electron to the point it doesn't just jump orbital, it jumps out of the chlorophyll, up here, and finally onto the electron transport chain. So, to sum up, this photosystem's main role is to harvest light energy so that we can concentrate it into the electron of this special chlorophyll so that we can have enough energy to be donated into the electron transport chain. Plants also have an electron transport chain. Let me show this in video so that you see some movement.
Thylakoid membranes contain specialized molecules that work together to perform the light reactions. Light is absorbed by protein pigment complexes called photosystems. There are two photosystems, photosystem 1 and photosystem 2. The photosystems transform light energy to chemical energy by exciting and then shuttling electrons from molecule to molecule in a chain-like fashion on the thylakoid membrane. This process is called an electron transport chain. Let's take a closer look to see how this process works. First, photons of light hit chlorophyll, a light-absorbing pigment in photosystem 2. Electrons in the chlorophyll are excited to a higher energy level. The excited electrons are passed to an electron carrier. Meanwhile, water splits and releases electrons. These electrons replace those lost at photosystem 2. The byproduct of this reaction is oxygen, which is eventually released into the air. The other products are protons, or hydrogen ions, which are released into the inside of the thylakoid, or lumen. The excited electrons move to the cytochrome complex. Some of the energy from the electrons is used by the cytochrome complex to transport additional protons into the lumen. The second electron carrier, a protein inside the lumen, receives the electrons and passes them to photosystem 1. These electrons have now lost most of the energy they gained from light in photosystem 2. Photons of light hit chlorophyll in photosystem 1 and excite the electrons again. The electrons are then passed to the third electron carrier. Finally, these electrons are either recycled or they interact with an enzyme in NADP+, the final electron acceptor of the light reactions, to form NADPH. Some of the energy from light is now stored in the reduced molecule NADPH. Some of the energy released from the transfer of electrons established a proton gradient across the thylakoid membrane. Protons that accumulated in the lumen diffuse into the stroma through an enzyme called ATP synthase. ATP synthase uses the potential energy of the proton gradient to combine ADP with inorganic phosphate to form ATP. In this way, the potential energy is transformed into chemical energy stored as ATP. ATP and NADPH now have stored energy from the light reactions.
Everyone's very safe. Yeah, so if we broadly look at this electron transport chain, several differences if we compare it to the cellular respiration. Number one, in photosynthesis, the electron transport chain comes with photosystems that contain chlorophyll. Number two, you see that the first thing that donates electrons isn't NADH. It is this photosystem that donates electrons from the chlorophyll. If you look at it broadly, you'll find that there are some similarities. In both instances, ATP is produced. But for what purpose, you'll see why in plants. But if you look at it broadly again, you notice that this electron transport chain is almost like the reverse of cellular respiration's one. In cellular respiration, NADH was the one that donated the electrons. And oxygen is the final acceptor to produce water. All over here, water helps to replenish any electrons lost. And NADP plus is the one that receives the electrons to form NADPH. So it's almost like a reverse process. Everything else in between is very similar. The idea of pumping protons from one place to another, and protons going down the ATP synthase. It's all the same. The main differences are the ones that we can broadly see in green on the screen. Okay, let's stop here today. I think I'll break nothing that much. Okay, so you go for a break. Lesson ends at 2, right? We will continue on the next lesson. In the next lesson, we will compare this electron transport chain to the cellular respiration. Okay, I'll see you at 2:30 in the lab, IS lab. Go and take a nap, a good nap first, before I see you again in the lab. Alright, thank you, class. Okay, thank you.